Well, hello. I know that the audience is joining us. Whilst you're settling down into your seats, I would like to welcome you all to this very special session on frontiers in thrombosis research. I think that we should be very proud that the United Kingdom is one of the global leaders in thrombosis research, and we have three eminent speakers who are international experts in their own right to bring us up to date into three frontline areas of research. So I'm going to welcome them all one by one. We will have questions after each talk, but if there are some amazing questions that require three of them to answer, then we will have a few at the end if there's time. So please put your questions in the chat if you have anything specific for each speaker. So I'm going to introduce our first speaker and welcome very warmly Professor Cheng-Hock, who is Professor and Consultant Hematology in Liverpool. He has had an illustrious career in organising things, and he's currently chair of the National Blood Transfusion Committee. He's just recently stepped down from being president of the British Society for Haematology, and he was academic vice president of the Royal College of Physicians. Despite all this administration, he does a lot of research, and he's had a long-standing interest in patients with sepsis and critical illness, and some of his work has led to spin-off companies. He is going to talk to us today about the new advances in understanding the pathogenesis of HITT and VITT. Thank you very much for coming today.
Thank you very much, Beverley, for that introduction. Can I just double check that people can see my first slide?
Beautifully.
Thank you. So that's a very generous introduction, Beverley, and it's certainly really, really important that that I sort of share my work with this audience and on the platform that also has Nicola and Suthesh as well. So I'm based in Liverpool, and my affiliations are to the hospital, the university, of course, a little bit still to the Royal College of Physicians. I also want to highlight that an important aspect of our work is linked through the Liverpool School of Tropical Medicine, particularly in its center in Malawi. So some of the work that I will discuss today will reflect on that. And as Beverley highlighted already, I've had a long-standing interest in hemostatic dysfunction, and in particular, the challenges that it poses to clinicians when you have that horrendous scenario that you see sometimes during critical illness when thrombosis happens at the same time as a bleeding potential. So it's extremely difficult for us who are clinical treaters to know what to do. And in a way, the recent reports of vaccine-induced thrombotic thrombocytopenia has shone a spotlight on it, but we've already had some insights into what's happening from the earlier experience with heparin-induced thrombocytopenia. So I'm just going to run through a couple of these things, and hopefully at the end present a clearer understanding of where we are today and where we intend to go forward. So just some representation of clots and potential for bleeding in the kind of semi-artistic way, but specifically we will discuss the mechanisms that link the two rather than actually cause them to be coincidental, coexisting processes. It's clear that in a number of these cases, there's a huge urgency about it, and we will discuss why that is. And essentially, a lot of these the presentations are largely in multiple sites over the body and therefore quite systemic in nature. And hopefully we can sort of share some insights about potentially what is in the future, what's in the horizon about ways to better improve the way we manage and care for patients that present to us in this way. So let's start with heparin-induced thrombocytopenia. And I'm not going to say a lot more about HITT beyond this, because some of the things that we will discuss later are informed by what we know about HITT. So HITT's been known about for a long time, certainly even before my consultant career began in Liverpool. And a key step was the identification of the role played by platelet factor 4. Now, platelet factor 4, as the name suggests, is derived from platelets, from the alpha granules in particular. And certainly, in my experience, is often sort of release or increase in the circulation as a result of a reaction. So we call it an acute phase reactant, very much like other molecules such as C-reactive proteins. So PF4 is probably evolutionarily old, and the way it comes to do its business is because of its positive charge. It's kind of a reaction to anything that's seen to be potentially foreign. So in this instance, in some individuals, heparin is recognized possibly as a foreign molecule into the body. So heparin, as you know, is negatively charged. Platelet factor 4 is positively charged. So the two come together really quite neatly. And for reasons we don't know and still continue to try and understand, some patients just form an immune response to this complex. So it's antigenic, and some will mount this antibody that can then bind to relevant receptors. And in platelets, of course, the idea is that the platelets get activated, it releases all its gubbins, many of which have procoagulant properties, and that kind of promotes that clotting potential. And of course, microparticles, which are released from the membrane, have that phospholipid surface, which is negatively charged, that acts as a suitable platform for clotting to occur. But in the last couple of years, we've started to understand that in HITT, the role of neutrophils is also very important. So the name, of course, when you talk about thrombocytopenia, just makes you think of platelets. And of course, clinically, we see a huge platelet drop. We don't see a huge neutrophil drop, but in a sense, underlying all this is the fact that neutrophils are activated, nets are formed, and I'll talk about nets in a moment. So when vaccine-induced thrombosis and thrombocytopenia became to be recognized, and Beverley and many other colleagues across the country were pivotal in highlighting this, we start to think, well, what is that immunogenic stimulus? And the reports have mainly been in the kind of adenoviral vectored COVID-19 vaccines, for example, made by AstraZeneca and Janssen. So we think that there's something about the adenoviral vector, and the AZ one is from chimpanzees, or potentially, or some of the composition material within the vaccine does seem to trigger PF4 to react. So maybe there's some degree of inflammatory response. PF4 then clusters with the viral capsid, and the hexon protein is the main kind of capsid protein on the adenoviral vector. And again, for reasons we don't understand, some individuals just start to make a lot of antibodies, and this whole thing sort of starts to get out of control. And of course, we then see platelets becoming activated in the same way as it did in HITT, but also the relevance of neutrophils again, and the release of neutrophil extracellular traps. So the publications, particularly by Andreas Greinecker, shed some light as to the role of neutrophils and nets in the VITT process, and a more recent publication involving in vivo mouse models has also highlighted that nets are particularly important to the thrombosis sequelae in this condition. So in a mouse model, if they block netosis, they seem to be able to reduce thrombosis. Likewise, if they have an antibody against platelet activation, they are able to abrogate the development of thrombocytopenia. So mechanistically, we think that an important trigger for NETs formation is actually cell death. Now, a lot of cells clearly can die, but neutrophils in particular, but if you remember, neutrophils often have several lobes. So when they release their nuclear content, they are particularly potent in getting out their DNA, the contents that are important for nuclear and cellular regulation, and they all come up in a kind of amalgam, which actually pictorially looks very much like nets, and functionally does seem to be able to trap microbes or pathogens. So evolutionarily ancient way of dealing with infection and bugs, but not only to trap them, but to kill them, because present within these strands will be enzymes that neutrophil elastase, myeloperoxidase, and a molecule called histones. Well, what are histones? Well, in some circles, histones are regarded very much as a very helpful molecule. You know, essentially, they bind to DNA, and DNA probably is about the length, is about my height, 5'10", if you unravel it all. And what histones do is it's like a spool that wraps it all into a tiny, tiny little speck, and so that a lot of DNA information gets tightly packed into the nucleus. So any disturbance or any disruption of the nuclear membrane will threaten the release of chromatin, DNA, and the release of histones. And what we know is that histones behaving outside the cell is really very different from being helpful within the cell. So some of the work from our lab earlier on now, about 10 years ago, has demonstrated that histones can bind to and can damage cellular walls. And if I point you to some of the figures on the left, these are in vitro experiments. So endothelial cells were incubated with FITC label histones, and you can see that the histones decorate the boundaries of the cell, nothing in controls. In vivo, if you infuse histones into mice models, you can see the lining of the vessel wall is again stained with histones, but not in controls. And something happens to the cell because calcium flux is disturbed. So diagrammatically, this is a simple way of just illustrating this very basic function that histones just punch holes into the cell membrane wall. And if it's small holes, perhaps there isn't much disruption in the ionic balance, but if it's large holes, the cells will have an influx of calcium and they will die. So again, maybe a quick way of remembering today's lecture is actually, that binding of histones to walls and the potential consequence of that happening. So thank you to Anish Kapoor. So in a nutshell, histones can explain some of these clinical problems that we see when thrombosis and thrombocytopenia happens at the same time. There seems to be a predilection when we infuse histones for the damage to be most apparent in the lungs. And we think it is because of the nature of that organ, because if you imagine that sort of outside-inside interface, the alveoli is important in the sort of first line of defense against invading pathogens, and neutrophils are very much in circulation in those capillaries. So histones and nets and stimulation of neutrophils to release nets is very important. But at the same time, if you stain for fibrin, it is very, very much evident in these mouse models of histone infused. but small vessels are also implicated. So you get large vessel clots, small vessel clots, as in the kidneys. The other finding is that histones will damage platelets and cause profound thrombocytopenia. And in the clinical cohort, some of which is described in the JAMA paper that we published, is basically if you take a critical care cohort of patients, and there are those whose thrombocytopenia can be explained by medication, and you exclude those that have DIC and you exclude those, there's still a significant proportion of critical care patients that have thrombocytopenia, usually isolated without any coagulopathy. And in these patients, they actually have very high histone levels, and prognostically, they do as badly as those who have DIC. So we think that histone-associated thrombocytopenia in critical illness is an important thing to consider. So let's move on now to VITT. So again, it's very common knowledge now, and the understanding has, of course, been held by a lot of UK hematologists, that problems with thrombosis happens, of course, in the venous sinuses, in the brain, but actually also in the artery. So not just in one vascular system. So some of these CT head scans, this is, for example, middle cerebral artery, internal carotid artery gets blocked by thrombus. The kind of hypoperfusion or the infarction that starts to happen. The same patient 24 hours later, and you see this soft area of infarcted brain. But also at the same time, in the same patient, in the portal circulation, there's clot, and in the pulmonary circulation, there's clot. So it's happening not just in arteries or in veins alone, but it's happening in several different locations, often ones which are very unusual, like cerebral vein sinus, like in the portal circulation. So some of these areas may be explained by the physiology in that system. They are kind of low pressure, low flow system, which might encourage certain types of prothrombotic potential to be played out more widely. This slide is just to highlight that, again, some of the things that I've alluded to and probably you know already. But if you look at markers of these things, for example, endothelial activation, ICAM1, this should read VCAM1, they are raised in VITT patients. We also ran controls, which are people who've had the AZ vaccine before and after, and we collected their blood. So it's nice to know it as a comparator. Inflammatory cytokines are hugely elevated, particularly IL-6 and IL-8. There is some Th1 and Th2 responses. And the interesting thing is that at the beginning of their illness, there is slight elevation of these particular cytokines that interfere, but actually longitudinally, they seem to drop away. And as we know, clinically, LDH levels are very high, indicating cellular damage. And if we look closely, I'm not sure if it's causally related, but we suspect it probably is. There's usually a lot of lymphopenia, which probably is from cell death. So we ran some NETs assays in these patients, again, using the relevant AZ controls, nothing in terms of formation of nets. And in VITT patients, what we do is we incubate their plasma with normal neutrophils, and those normal neutrophils get activated and extrude nets, which are stained for the DNA and show up quite vividly. So quite clearly, no second guessing nets are there. And in Greinicker's paper, when he looked at the histology from the cerebral vein sinus thrombus, there's co-localization of neutrophil elastase, DNA, histones, so all tightly packed within that thrombus. So what about histones? Well, they are, again, surprisingly, not surprisingly, very high because of the extent of cell damage, the extent of mitosis. And if you run this plot where you look at the levels of circulating histones against these various parameters, there is that kind of very strongly positive R value or correlation that you can see here with the things that we know about, but also in relation to those clinical parameters that we routinely measure. So high histones, lower platelet count, high histones, lower fibrinogen level. D dimer, of course, is high as characteristic in these patients. So let us think about whether histones are just there in a kind of epiphenomenal way. You know, maybe it's just release, and yeah, it's a good marker, but does it tell us anything more? And for a lot of us working in the critical care setting, finding tools that can help the clinician predict the clinical course or predict adverse events can be helpful. Maybe it makes you look at the patient more carefully on the day-to-day or hour-to-hour basis, but certainly in terms of prognostication, we can tell the difference. So in the VITT patients, if you categorize them in terms of survival at 28 days, those that did okay really had barely noticeable histone levels, whereas the non-survivors clearly were leaping out in front of you. And if we run odds ratio analysis, we seem to find that a histone cutoff of 30 is important. In the lab, it's important because we can demonstrate that concentrations less than 30 don't seem to be too cytotoxic. Above that, it starts to be quite damaging. So clearly histones beyond 30, high p-value, odds ratio for predicting mortality, even when we adjust for other variables. And this is, you know, quite a clear difference in the outcome. So if you can read that from where you're looking, you know, very high area under the curve for circulating histones that are high, and very high odds ratio given as a numerical number here. So, of course, it made us look again at some of the work we've done in the past as to how to explain a lot of these findings. And as many of you know, one of my pet interests is in how coagulation is disseminated. We've started to make a lot of headway in understanding how histones can contribute to thrombin generation in vivo. So I point you to this paper that was published in Blood in 2020. And then there was this paper where we could demonstrate clinically that if you measure patients on arrival to the intensive care and their NETs forming capacity, you could predict the development of DIC and of mortality. I'm just going to talk through a little bit of what we found in this paper because it may give us some further insight into the coagulopathy that is seen in sepsis, but also in VITT. So the classical way of us thinking about thrombin generation in vivo is the formation of this complex called prothrombinase. And we know that prothrombin will convert to thrombin, particularly in the it's when factor Xa cleaves prothrombin in the presence of calcium, in the presence of phospholipid. And all that means is that the reaction can happen at the membrane level. You know, phospholipid is the anchor to allow the co-localization of things to make the reaction run faster. And it's catalyzed and accelerated by the presence of cofactor, factor 5A. So this is classical prothrombinase. What we've been able to demonstrate is that actually you still get this reaction with the same molecules involved in the absence of factor 5A and in the absence of a phospholipid membrane. So yeah, okay, this is another prothrombinase. We call it alternative prothrombinase. But we think that the fact that it can happen, a reaction that can happen away from the membrane surface, indicates that if histones are in the circulation and it collides with prothrombin, there's a chance that this will kick off. So it's an opportunity for thrombin generation to be disseminated away from the membrane surface. So in vitro, we can show that if you add small doses of histones, you can correct the clotting abnormality caused by factor 8 and factor 9 deficiency. So you kind of almost normalize the clot. We also injected it in hemophilic mice, and basically low dose histones, which doesn't damage the cell and its environment, will shorten the clotting time. Histones can be neutralized by heparin, again, a charge mechanism. And you will see that in the presence of histones combined to heparin, there is no shortening of clot time. So we think that actually in vivo, this is happening too. So if we give high doses of histones into mice, you will get a DIC phenotype, all the things we know about as clinical and laboratory people. TAT levels go up, DIC score also goes up. We think that the alternative prothrombinase is happening in that situation, because if we also infuse this, what we call a decoy molecule, we just create prothrombin fragment 1+2 and infuse it. It will compete out with histone binding, and in co-infused animal studies, it will reduce the TAT formation, showing that a lot of the histone-induced thrombin generation is by this alternative prothrombinase route, and likewise for changes in fibrinogen levels. So let's go back to VITT and the presence of, or not, of disseminated intravascular coagulation. I think it's fair to say that that a lot of us have perhaps different perspectives of what DIC might mean, and I think it's colored by the kind of patients we've looked after, whether it's predominantly thrombotic, predominantly bleeding, or a bit of both. And many moons ago, I was lucky to be involved with trying to get a grip on it and standardizing how we define it. And crudely enough, we rely on things that are easy to do. And so we compile the scoring system, give it a number that means something significant. And I've shown that actually, if patients reach that kind of number, it is more likely that they will die. So if we look at VITT patients, actually, although the prothrombin time is seldom that elevated, but because of their D dimers and the fibrinogen in their platelets, they actually get labeled using this ICH scoring system as having DIC. And the ones who have DIC, no surprise, are the ones who are likely to be doomed. But if we also look at their plasma, so an indication of what's happening in their circulation, we can stain for fibrinogen fibrils that are slightly disturbed using this sort of thioflavin staining. So in our patients, their plasma seemed to light up, indicating the presence of some soluble clots or microclots, or whichever you prefer to use. And we know, as has been demonstrated by others too, that there is fibrin within the tissue. So in terms of how you define DIC, certainly there is dissemination of a coagulopathy, and there is thrombus happening in places that we don't think is healthy for it to be in. So let's try and wind down. So in a nutshell, really, I think to try and make it simple, really what we think is happening in critical illness is this propagation of thrombin generation that is very much histone dependent. And of course, when histones cause cell death, the cells release histones, so you get this cyclical, potentially vicious cycle of happening events that may get out of control. And of course, interrelated to that is the formation of nets. We know that histones will cause neutrophils to release nets, and likewise, when nets are released, they contain histones which can do their business. And platelets can activated platelets can stimulate nets, and likewise, vice versa. So in a nutshell, all these things are connected, and all these things can make thrombosis and thrombocytopenia occur at the same time. So in drawing this talk to a close, I hope, even though I haven't talked too much about heparin and HITT in particular, some of the things that are happening in these patients are not dissimilar. I think we're better at recognizing HITT earlier, and that's why we don't see some of the consequences. Although in the earlier days, you know, DIC and HITT were interrelated in some patients. So perhaps one of the take-home message is don't just think about platelets, think about neutrophils and what they do. The platelets do bind to the nets and cause a lot of problems, of which we've seen in VITT with this kind of profound manifestation in D-dimer, platelet drop, and falling fibrinogen levels. So in summary, the take-home message again is, you know, think of these things as the body's sort of alarm response. You know, it sort of senses for whatever reason that this particular vaccine or heparin may be threatening, and so it's mounting this huge reaction to try and neutralize it, and as a consequence, increased thrombin generation happens in vivo. Thrombin generation then plays out usually in localities, which is mediated by nets because it sort of acts as a scaffold to trap all the coagulation for important molecules, and histones are particularly relevant in making that happen. So clinically, where do we go? I mean, I don't think we're, we're not proposing any randomized clinical trial at the moment. Luckily, VITT is not that obvious now, but I think moving forward, we would want to consider whether markers such as histones or nets may be useful early on. To make them useful, they need to be quick, simple, easy to do in the lab. We're not quite at that stage, but you know, clearly technology is advancing leaps and bounds these days, and there's no reason not to be hopeful. And of course, that also points to this sort of diagnostic therapeutics, you know, the link between finding a biomarker that we think is functionally relevant. This whole gray area of immunothrombosis, we are still lacking good drugs. You could think about anti-nets type of treatment regimes. DNase has been proposed. Heparin has been proposed. The problem with heparin is, although it does dissolve nets, it will aggravate the bleeding side of thrombocytopenia. But there is a kind of non-anticoagulant heparin that is around. Whether it's commercially viable is another matter. So put it this way, there are lots of discussions happening for all the right reasons. We hope that they will add to our armory of treatment opportunities as we move forward into the future. So with that, thank you for your attention. And I am really happy to address any questions that you might have.
Well, thank you very much, Cheng-Hock. And that was spectacular and very, very current. I just have one question. We're running a bit over time, but I really wanted to know about neutralization of histones with heparin. So does it vary between the different heparins? Have you looked at fondaparinux?
I think the good news is that virtually almost every heparin, certainly those with anticoagulant properties, will have antihistone properties. So yes is the answer. Fondaparinux included. Low molecular weight heparin or unfractionated heparin will do.
Okay. Well, thank you very much. That was absolutely wonderful. I'm afraid we have to move on now because of the time. So thank you very much. And I'm moving on to our second speaker, who specifically is going to talk about advances in fibrinolysis. And Nicola Mutch has been well known for many years for her work in fibrinolysis, and she has now got a personal chair in Aberdeen since December 2021. And Aberdeen is the place for fibrinolysis in the UK. We've got a long backstory of people like Nicola and Bruce Bennett who worked there and made major advances in fibrinolysis. So it's a real joy to have Nicola here. She's actually on holiday in Nice and is breaking her holiday to come and talk to us. So Nicola, thank you, and over to you.
Many thanks, Beverley. It's a lovely introduction, and it's really a pleasure to be here to speak to you all. And it's lovely also for you to mention how fibrinolysis has really a home in Aberdeen, and we hope that continues for a very long time. So just to confirm before I move on, everyone can see my slides. Let me know if you cannot.
We can.
Fantastic. Thank you. So the fibrinolytic system is in some ways viewed as the poorer cousin, if you like, of the coagulation cascade, that pathway downstream that people don't really focus on too much. But I think in the past few years in particular, it's really become apparent to other individuals, more coagulation or platelet-based researchers, how important the fibrinolytic system is in dictating that overall clot stability and provoking thrombosis, and also on the counter side and taking part, obviously, in bleeding pathologies. So this is an overview, a simplistic overview of the fibrinolytic system. And you're going to see here the activation of plasminogen. So like its counterpart, the coagulation system, the main proteolytic enzyme circulates in a zymogen form, so a quiescent silent form in the circulation. And this is activated by plasminogen activators, namely TPA and UPA, although there is roles for other plasminogen activators. These act on plasminogen and form the active enzyme plasmin. And it is plasmin that can then degrade that fibrin network into fibrin degradation products, which can be cleared from the circulation. You'll see here this sort of hump shape here. What this is to represent is the fact that fibrinolysis is relatively poor if it doesn't have a surface to take part to participate on. Fibrin is considered the classic surface, and that is because in the absence of fibrin, TPA is a relatively poor activator of plasminogen. And actually the activation is stimulated over 1,000-fold in the presence of the fibrin as a binding site for both plasminogen and TPA. But UPA is fibrin independent. So what does UPA use? Well, UPA tends to be involved in terms of a cellular surface. So there is a receptor called UPAR that is present on a number of circulating cells in the vasculature, and UPAR will localize UPA to the surface. There's also, and I'll come on to this later in the talk, binding sites for plasminogen on these cells. And as a result of that, we then see the surface-mediated activation of plasminogen to plasmin. Not surprisingly, the system is under regulation by a number of different physiological inhibitors. These include SERPIN inhibitors, again, classically the same as seen in the coagulation cascade. PI1 is the dominant inhibitor of the plasminogen activators, so it will inhibit both TPA and UPA. And alpha-2 antiplasmin will directly inhibit plasmin. When the system becomes overwhelmed, there is other inhibitors that can step in and help take control of the whole process. And these include C1 inhibitor and protease nexin 1, which is released from platelets, which is not shown on the slide. I must modify it. In addition, down here, you'll see an inhibitor that's complex in its nomenclature. It's called thrombin activatable fibrinolysis inhibitor. It's also called carboxypeptidase U for unstable, or plasma carboxypeptidase B. So many different names, but essentially what this inhibitor does is it is activated by thrombin in the presence of a cofactor thrombomodulin, and to a lesser degree, but maybe also important, by plasmin itself. Instead of binding directly to an active protease, as do the SERPIN inhibitors, TAFI will downregulate fibrinolysis by removing C-terminal lysine residues from the fibrin surface, therefore downregulating binding of plasminogen and TPA to that fibrin, and thereby attenuating the whole fibrinolytic pathway. You'll see other players in here, and much as I'd love to talk about them, we don't have time to go into all of them today, but there is, I'd like to mention this one particularly unusual SERP in here, PI2, which we really have a lot to learn about, because PI2 is largely intracellular in origin, but becomes highly upregulated during pregnancy. So it's really the only time that it is found in plasma. And there's also potentially a role for the contact pathway in terms of regulating the fibrinolytic system. But again, I won't focus too much on that today. Why are we interested in fibrinolysis? Well, it's well known that increases in a number of the inhibitors that I've just spoken about, PI2 and alpha-2 antiplasmin, can cause an increase in both arterial and venous thrombosis and peripheral arterial disease in the case of PI1. And these are predominantly the ones that have been studied. TAFI has also been studied, and there are reports in the literature that increases in TAFI can be associated with an increase in arterial and venous thrombosis. However, the literature is somewhat confounded by the fact that there was issues with the specificity of assays that were used in a number of these different studies, and perhaps that this information must be revisited at some point. But there's some indication that there could be a link between increased TAFI levels and a predispensity to thrombotic conditions. Factor 13 is a really important enzyme because we consider it to be part of the coagulation cascade, but nonetheless, factor 13 actually participates very widely in fibrinolysis, and it does this by becoming activated, again by thrombin, during the whole coagulation process. But when it becomes activated by thrombin during those initial stages of clot formation, factor 13 can cross-link inhibitors, predominantly alpha-2 antiplasmin, into the forming clot. And as a result of that, we get a clot that's very resistant to fibrinolysis. The converse of the fact that increase in the inhibitors of fibrinolysis are associated with thrombosis is the fact that a deficiency in these factors is associated with bleeding disorders. And so while rare, PI1 deficiency or alpha-2 antiplasmin deficiency are associated with a mild to moderate bleeding disorder. Interestingly, a deficiency in factor 13 is associated with a very severe bleeding disorder when the mutation resides within the A subunit of factor 13. Again, I took all of this out of my talk because I decided I didn't have time to go into it all today, but factor 13 is a very complex enzyme. It's not a serine protease as most of the other coagulation and fibrinolytic factors are. It's a transglutaminase enzyme. And in plasma, factor 13 exists as a heterotetramer of 2 A subunits and 2 B subunits. The A subunit houses the catalytic activity, so it really performs all the functions of factor 13, and the B is a carrier subunit. So perhaps not surprisingly, deficiency in factor 13B is associated with a more mild or moderate bleeding disorder. And to this date, we still don't know the origin of the factor 13 subunit that is present in plasma. There's a cellular source, and it was originally thought that it came from platelets, but this does not seem to be the case. Factor 13B is produced in the liver. And interestingly, this transglutaminase enzyme is also associated with acquired bleeding deficiencies, and they can be particularly difficult to diagnose, and I'll come back to that later in the talk. But of course, it's not quite as simple as all of that, because I've mainly up to this point mentioned the protein players, both the pro and the anti-fibrinolytic proteins that are involved in fibrinolysis. But what the previous diagram did not show is the cellular involvement, and that's really going to be a focus of my presentation. I could have talked about all of the cells and their role of regulating fibrinolysis, but we've had that wonderful presentation by Professor To on the role of neutrophils in modulating coagulation and potentially downstream fibrinolysis in various different disorders. So I thought today I would focus again instead on platelets. But interestingly, just to point out before I leave this slide, that red blood cells are potentially also involved in terms of dictating that clot stability by a variety of different mechanisms. So we all think of platelets as being integral to hemostasis, but perhaps more so in relation to their involvement in coagulation. And that's not surprising given the fact that when platelets become activated and expose aminophospholipid on their surface, such as phosphatidylserine, they will bind the coagulation factors through their glutamines and act as a site of thrombin formation. Coinciding with that thrombin formation is the fact that platelets harbor a number of receptors. And again, we won't go into all of those today, but I've illustrated on here alpha-2 beta-3, which is classically considered the fibrinogen receptor on platelets and will bind both fibrinogen and von Willebrand factor. And as a consequence of fibrinogen binding to alpha-2 beta-3, it then acts as an anchor to draw together two different platelets, and also as a point of stability, so anchoring the platelets into that forming fibrin network. The platelets then play a really important role in dictating the overall structural integrity of the clot, because that binding of fibrinogen causes extra to intracellular signaling response, which through interactions with the actin cytoskeleton of the platelets will then result in a tightening of that fibrin network. And that's illustrated here in this diagram that we've got. This is a clot that's formed under shear stress in a microfluidic chamber. And you can see the fibrin is formed in red due to a fluorescent labeled fibrinogen, and the platelets are labeled with a lipophilic dye DIOC6. You can see that the fibrin is immediately adjacent to those platelets, again indicating the importance of this anchoring of the platelets into the fibrin network through interactions with fibrinogen. There has since been other receptors for fibrinogen identified on platelets, including GP6, which there's been some lovely work on, and actually this is the only receptor in which fibrinogen has been shown to directly activate the platelets. So if we look at this in more detail, and Claire White from my laboratory was instrumental in developing a model that we were able to monitor fibrinolysis under flow in a microfluidic system. And this uses whole blood, because part of our limitation was the fact that most of the earlier assays that we had performed, which, don't get me wrong, were exceptionally important and identified many of the main protein players in the pathway, they lacked the cellular components of the system. So we need and require a more complex system in order to be able to take into account the influence of these cells in fibrinolysis. And what you can see here is that if we look at the clot that is formed, again, this is similar to the previous, so the platelets are in green and the fibrin is in red, and this is at time 0. We then infuse TPA into the blood. The reason we have to infuse TPA is because there's a very overwhelming balance of the fibrinolytic inhibitors in this case, and as a result of that, TPA will largely come from the cellular sources such as the endothelium. So we include TPA, and as we increase the concentration of TPA, as shown in this bar chart here, we can increase the rate of fibrinolysis. But what you will note here is that it is the fibrin that's immediately adjacent to the platelet surface that is most resistant to degradation. So why is that? Platelets, again, classically considered to be important in coagulation, but many years ago, platelets were identified as the main circulating pool of PI1, the SERPIN inhibitor of plasminogen activators. And PI1 is actually stored in many different, and synthesized by many different cells. And you can see here it's synthesized by endothelial cells. Hepatocytes are able to synthesize it, but importantly, it is synthesized at the megakaryocyte stage and packaged into the alpha granules of platelets. Again, I don't have time to cover this today, but we also know that adipocytes are an important pool of PI1, and in fact, in obese individuals, that pool of PI1 can contribute to the plasma pool. So it is obviously being secreted from the adipocytes into the circulation. This is a very complex slide, but produced by Gail Morrow in my lab, but it just shows the discovery of PI1. and really a timeline of how our understanding of this complex serpent in the regulation of fibrinolysis and the interplay in other processes such as cardiovascular disease, which is very well known now, but also more recently in metabolic syndrome. And I will come back to that if I have time later. But I was also focused just at the end of my talk on the role of PI1 in various viral diseases. So just to illustrate that again, we know that elevated PI1 is associated with cardiovascular diseases, bacterial and viral infections, and this metabolic syndrome, which is a syndrome that's comprised of multifactorial syndrome that's comprised of an increase in hypertension, obesity, and type 2 diabetes. These images here are produced under a confocal microscope in the absence here of PI1, a neutralizing antibody to PI1, and the presence of a neutralizing antibody here. So you can see here that when we take out PI1, we dramatically accelerate clot lysis, and this clot was gone almost immediately, whereas we're still seeing residuals of this clot here. I'll try to play that for you very quickly again. And again, you'll see how rapid it is when we take PI1 out of the equation in this setting. And we see that PI1 and its cofactor vitronectin, which it circulates in complex with, are localized within that fibrin network. Work from Gail Morrow, which was funded by Thrombosis UK and the British Society of Haematology and the British Society of Hemostasis and Thrombosis, was able to demonstrate that PI1 is retained on the activated platelet membrane. And we found it located in areas that were rich in phosphatidyl serine, which is very interesting to us because this had commonly been associated as areas for coagulation factors, but not for fibrinolytic factors. And indeed, PI1 was thought to be part of the secretome, but not retained on the activated platelet membrane. And we were able to show through blocking alpha-2 beta-3 with tyrofibrin or blocking fibrin polymerization that the interaction of fibrin and alpha-2 beta-3 were very important in being able to retain that PI1 on the activated platelet membrane. So as I mentioned, PI1 is associated with cardiovascular disease. And importantly, I'm going to point out the second point here, that plasma PI1 is at a relatively low concentration, but as we age, our plasma levels of PI1 increase. PI1 is also under circadian rhythm, and we're probably still in our infancy of understanding all of that, but it's very well known that it's PI1 is at its highest early in the morning, and that is associated with the highest peak of cardiovascular events and strokes. We also know that PI1, if it's overexpressed in animal models, results in development of atherothrombosis and venous thrombosis. And PI1 is found within atherosclerotic lesions. So some important work that's been done recently by the group of Professor Diana Gorok has been identifying the role of hyperfibrinolysis in STEMI patients, myocardial infarction patients, and its association with acute coronary syndrome. So they published a nice paper in 2018, which showed that hyperfibrinolysis is a risk factor for recurrent cardiovascular events in STEMI. And this is despite PCI treatment and antiplatelet therapy. But we also know that if I go back to the diabetes and the metabolic side, that diabetes is associated with a heightened risk of cardiovascular events, and also is associated with increased levels of PI1. PI1 also correlates with insulin resistance, and as I mentioned before, is a risk factor for metabolic syndrome. So in a sub-study of the risk PPCI study, we have looked at patients that are non-diabetic and diabetic, and not surprisingly, there is differences such as a difference between BMI in these groups and hyperlipidemia. There's also increases in C-reactive protein and fibrinogen, so these are more elevated than diabetic patients. But there is also a dramatic increase in the lysis time, so showing that the clots formed from these individuals are more resistant to lysis. And I should point out that this was done using a point-of-care instrument called a ThromboQuest on non-anticoagulated blood. But interestingly, if we look at thrombin potential in these individuals, it's not different between the non-diabetic and the diabetic cohort, indicating to us that that difference does not lie in changes in the coagulation cascade. But what we then see is that if we look more closely at the fibrinolytic parameters and we look at the plasmin endogenous plasmin potential, it's a mouthful, we see that there is depressed plasmin generation in diabetic patients, and this correlates with a slower lysis time in these individuals. And if we dig down deeper and look as to why this is potentially could be, we find that there is elevated Pi1 in these individuals. So the hypofibrinolytic response that is observed is a result of these high Pi1 levels. So I mentioned earlier the bacterial and viral infections, and we all know how quickly COVID came into our lives, but almost as quickly we recognized that while this was a respiratory disease, it was also associated with thrombotic complications, and there was dysregulated fibrin deposition both in the lungs and systemically in severely ill COVID-19 patients. And this is due to the immunothrombotic complications that Professor Tao has touched upon already, which I won't go into in detail, but there is a direct relationship between the hemostatic cascade and the inflammatory cascade, with upregulation of many coagulation factors, changes in cells with becoming more prothrombotic, including the activated neutrophils, monocytes, and platelets, and potentially also changes in Pi1 and tissue factor. So we had a look at severely ill COVID-19 patients and looked at their rates of plasmin generation compared to in the grey healthy individuals or in the blue individuals with pneumonia but non-COVID related pneumonia. And you can see that there is depressed plasmin generation in the COVID-19 cohort of patients. And in fact, this translates into a defective lysis time in these individuals as well, so slower lysis. We see changes in the fibrin network, which can again be associated with a more prothrombotic phenotype. But Pi1 was shown to be dramatically elevated, not even just in relation to healthy individuals, but also in relation to just pneumonia patients that were not COVID positive. This also was shown in terms of both Pi1 antigen and activity, and was associated with an increase in Pi1's cofactor vitronectin. So when we looked more closely at the ability of these clots to lyse, we compared two drugs, thrombolytic drugs that are available on the market, tenecteplase and alteplase. And these drugs differ significantly despite them both being variants of TPA. Alteplase is a recombinant form of TPA that is not as genetically modified, if you like, as tenecteplase, and that tenecteplase is resistant to Pi1, has a higher degree of fibrin specificity and a longer half-life. And you can see here, if we stratify these COVID-19 patients into low, medium, and high Pi1 levels, the difference in the lysis time between alteplase and tenecteplase. So tenecteplase is able to overcome the hypofibrinolytic response in these COVID-19 patients. And indeed, there's a correlation between the concentration of Pi1 and the delay in alteplase lysis, but not tenecteplase lysis. So tenecteplase is more effective at degrading clots in these patients. And in fact, there is a current trial in the UK, the TEST trial, looking at the efficacy of tenecteplase versus alteplase in acute ischemic stroke. And I think this will be very interesting to hear about. But I should point out from this slide that it is not just COVID-19. Pi1 is elevated in many other diseases, including sepsis and DIC. Other fibrinolytics inhibitors are also present in platelets, and I don't have time to go through that today. But very briefly, just towards the end, we know that not all platelets behave in the same way. And quite early on, when I was still a PhD student in Professor Booth's laboratory, we found that platelets were able to stimulate lysis when lysis was induced with single chain UPA. And since then, a body of work has unfolded for us to be able to look at these profibrinolytic properties of platelets. And what we can see here, this is the rate of scupa activation, both in solution or on the surface of platelets. So it's significantly augmented on the surface of platelets. And this is perhaps a surprising observation, given the fact that we've shown you how antifibrinolytic platelets can be in terms of their Pi1. But we know that there is reciprocal activation of plasminogen and scupa on the platelet surface. And my postdoc, Professor Claire White, has worked extensively on this, and we've been able to show that platelets can stimulate plasmin generation. And they can also act as a sink for plasminogen within the site of the clot. And these are some images similar to what I showed previously, but we're looking at localization of plasminogen in the thrombi that are formed under shear stress. And we find that a pool of that plasminogen is directly localized on the platelet surface. Interestingly, if we think about where these procoagulant platelets are that are active, they actually encase the fibrin. So the spread platelets are formed within the fibrin network, but the procoagulant platelets are on the periphery of this fibrin network. And you can see here, again, these procoagulant platelets that have plasminogen exposed on the surface. We've now have a more deeper understanding of what's going on through our collaboration with Professor Lindsay Miles at the Scripps Institute in California. She had identified a novel plasminogen receptor on the surface of monocytes, and we've now shown that this same receptor is present on the platelet surface and indeed is augmented post-activation of the platelets. So therefore the platelets will then be able to drive the profibrinolytic and the antifibrinolytic responses. And really, we're still trying to develop a deeper understanding of how this is all taking place. But we do know that these things differ according to shear stress. And if we look at thrombi that are formed under low shears, mimicking that in the venous circulation, we see that there is platelet deposition, but there is more fibrin deposition under low shear, and the fibrin forms as clouds around these platelets that are formed in the venous circulation. Whereas in high shear, what you can immediately see is that the platelets form almost as threads or pearl necklaces, I like to think of them as, but there's significantly less fibrin deposition around these platelets. But this also translates, and that this is work done with David Newby in Edinburgh, where we have looked at incorporation of fibrinolytic proteins in thrombi formed in the Badaman chamber. And we see significantly that there is a dramatic increase in the amount of Pi1 in thrombi that are formed in high shear, that's shown in the black here, compared to low shear, making them very resistant to fibrinolysis. And you'll see that there, in contrast, there is not such a significant difference, but there is also less plasminogen present, so less ability to be able to generate plasmin in these high shear thrombi. As a result of that, this translates into reduced lysis of the high shear thrombi, and there's a correlation with D-dimer. So it brings me on to just my final couple of slides. Why is our understanding of fibrinolysis so limited in this current day and age, and our translation into what's going on clinically? And really that is because we're really in our infancy in having appropriate tests to be able to diagnose fibrinolytic disorders. So we classically look at endpoints in the process, whether that be a D-dimer, as I pointed out on the previous slide, fibrinogen itself, soluble fibrin degradation products, or evidence of plasmin alpha-2 antiplasmin complex. We have made some advances more recently. And I know thromboelastography is very long-standing, but it's only just recently been used for fibrinolytic disorders. And this interesting data here from Mikkelsen et al., which was produced in 2020, shows an individual that's with trauma-induced coagulopathy that has arrived in the ER and has got a very defective fibrinolysis, and that's this channel here that is shown on the Quantra machine. Once they've been treated with TXA, their fibrinolysis is normalized. So perhaps we need some new diagnostics tests which will help drive our understanding of the relationship between our fundamental knowledge that we have of the fibrinolytic pathway and how this translates into clinically. And it just remains for me to thank everyone involved in all these various studies and all the funders. And thank you again for the very kind invitation to speak today.
Well, thank you very much, Nicola. So fibrinolysis remains strong in Aberdeen. We haven't got much time
Oh,
now.
sorry.
Would you just like to comment on the fact that we ought to be thinking a little bit more about some of the factors in coagulation fibrinolysis being like cytokines? For example, thrombin activates white cells, platelets, endothelium, it does this to this cell or that to that molecule. Is Pi1 a similar molecule, do you think?
A very similar molecule, and the fact that there is I didn't present the data on the relationship between Pi1 and cytokines, but it's very inextricably linked. And, you know, I think
it's part of the acute phase response, isn't it, really?
It is. That's exactly, and that's why we see it upregulated in so many bacterial, viral infections, etc., and in those sterile inflammatory states, such as diabetes and atherosclerosis and stroke. So really, I think we need to be thinking more about these interactions. You know, that's the key into our understanding. It was very important that we took our understanding of the individual proteins and how they all function, but now we need to pull it together and also think about the role of cells.
And thank you very much for the beginning, because you explained fibrinolysis and it's so poorly taught everywhere. So that's much appreciated. We need to let you get back to the sunshine and the beach. So thank you very much.
Thank you. Thanks very much.
Bye. And so now we're moving on to our last speaker, last but not least. I have done a lot of work with Dr. Suthesh Sivapalaratnam with the ISTH meeting in London. He was a key member of the local organising committee. He has a big clinical practice in haemophilia, one of the largest in the world, and he looks in particular after the paediatric service, and he's genomic lead at Barts. He's also got a company. He's chief medical officer of a company called Blood Counts, and he's done a number of consortia. He's got a number of grants, and he's got a number of publications. But he is particularly well qualified to talk about thrombogenomics. So thank you very much for coming today.
Thank you, Beverley, for the very kind introduction. And it sounds all very impressive, but compared to you, that's obviously still nothing. Thank you, everyone, for listening to the talk. And here is a nice view of, if you look out of the day unit at the Royal London, where I'm based a lot, and you can see on one side the city coming close to us. But also in this picture, you see very closely our biomedical campus. And I'm very fortunate to be part of that development. So today, I'll be talking to you about thrombogenomics. So what are the developments in genomics and genetics in thrombosis? And there's quite a few excitements going on. I've been in the field of genomics for quite some time now. And it's really nice to see how the kind of academic and scientific part is coming close to clinical practice. I'm very fortunate to be one of the consultants at Barts, and I hold an affiliation at QMUL. And a lot of the science which I'm presenting here has been made possible by the NIHR. So what will I talk to you about? So I'll talk about a little bit of the background of thrombosis. This is things that you probably know, but just to land you softly, then I'll go into genomics in thrombosis. So a lot of the things in thrombosis and the traditional risk factors were discovered through genomic research. I'll talk to you about the latest genome-wide association studies and the resulting polygenic risk scores. And I'll also talk to you about how this could implement our clinical practice and where it could be useful and where more work is needed. And finally, I'll show you some data on a really exciting recall study we're doing. And it's a bit of a plug for the study, hoping that you might be interested in contributing. So as you all know, there are numerous risk factors for thrombosis. And the standard risk factors are age, obesity, family history, obviously a previous DVT, recent surgery, hospitalization, and prolonged immobility. But now, recently, we also know that cancer can be a risk factor, chronic disease, and as recently shown, obviously, with the pandemic viruses, such as SARS-CoV-2, have been a major contribution in the risk for thrombosis. So these risks can also be quantified. So one of the major studies, which, as Beverley said, the UK has been on the forefront of, is the UK Biobank. So the UK Biobank is a large cohort of men and women between the age of 40 and 69 years who have volunteered to do this research. And this cohort, this prospective cohort, was initially recruited between 2006 and 2010, and it contains about 502,000 people. And if you look at this cohort, this is data published in Nature Scientific Reports, you can again see the different adjusted hazard ratios for venous thrombolism events. And some of the risk factors, like we discussed before, are here. So you can see that age is a risk factor, and with an increase in age, there's a higher risk of thrombosis. You can see the factors like BMI. So again, a high BMI of between 35 and 40 seems to be a clear risk factor for venous thrombotic events in this big cohort, and other things like smoking come quite clear. So here you can see the risk by hazard ratios up somewhere for the major factors between 2 and 4. As I said, family history is also a clear risk factor for thrombosis. And now, because I talk about genetics, it's very easy to just assume that that's all shared genetics. The estimates are that genetic risk factors contribute to 30 to 60%, but I'm very much aware that that will be shared environment and shared environmental factors, and some of the factors we discussed previously, like lifestyle, BMI, smoking, unfortunately also cluster in families, and part of that will be genetics. And if you want to pin that down with a number, papers published about a decade ago showed, and this is something we know, that once you have a sibling with a VTE, if there's 1, your ratio compared to the baseline goes up by 2. If you had 2 affected siblings, your risk goes up by a 50-fold, especially if that's a young age. So as I said, that reflects the risks coming with families. So when we talk about genetics in general and genetics in thrombosis, how do we even study that? So the standard way of studying that has been family studies. And through family studies, we found in the past the quite severe disorders. Later on, about 5 to 10 years ago, we went on to candidate gene studies. And more recently, we started doing genome-wide association studies. The technology for these studies has progressed massively, and met with progression of the technology, have we been able to type deeper and deeper. So when I started doing my PhD about 15 years ago now, we were very interested if we could look at a few variants in candidate genes. Later, towards the end, the SNP arrays came, where we can type about 50,000 initially, but now about 800,000 genetic variants on one chip. That wasn't good enough for the genomicists, so we moved on to whole exome sequencing, where we can sequence the variants of the 22,000 coding genes, which also make proteins. And now we even do whole genome sequencing, and those can be used for association studies. But the big question remains, what is the usability of all of these technologies in medicine? And I think there is some usability. So I'll talk to you in more detail about polygenic risk scores, of which 2 studies have been instrumental, the UK Biobank and the Million Veteran Program in the US, and how we can use this knowledge in NGS diagnostic platforms. So again, when you look at this figure, so here on the x-axis, you see the frequency of an allele or a variant, and on the y-axis, you see the penetrance of the disease. So we started with the low-hanging fruits in genetics. So if you have a variant which has a high penetrance and is very rare, then that normally tends to run in families, and you see Mendelian disorders. So in the hemostasis perspective, for instance, Glanzmann's, Bernard-Triet's are like clear versions of high penetrance and rare variants. If you want to study common variants with a low penetrance, that's where the GWAS studies and the association studies were most useful. And now, as time progresses, we're going down and down the other bubbles. And it's also more clear that this landscape is not this straightforward. So the inherited thrombosis, we all know from a clinical practice, are protein C, protein S deficiency, antithrombin deficiency, factor V Leiden, heterozygosy or homozygosy, and the prothrombin variant. And a lot of these deficiencies and protein deficiencies were picked up by studying rare cases or rare families. We also know, if you look at this review from the New England Journal in 2001, that none of these variants are very black and white. Obviously, even in populations which are healthy, people will have variants in these genes, will have reduced levels, but as you go into patient cohort and enrich for thrombosis, the percentage and the frequency of variations in these genes and variants increases. So in a healthy cohort, as you can see, for instance, for a antithrombin deficiency is 0.02%. If you look in unselected patients with the thrombosis, it goes up to 2%. And if you then look in selected patients, and by selected, I mean young, relatively young thrombosis with the family history of VT or recurrence, you can see that the frequency of variants and abnormalities in this protein increases. So that was how we looked at Mendelian disorders, but a lot in the past years have been around genome-wide association studies. So just to recap for those who might not know, genome-wide association studies are not complex. So on one side, you define a group of cases, and on the other side, you have a group of controls. And case control studies, we've done those forever. But what we do in the genome-wide association studies is that we type the cases and the controls. And by typing, I mean we look at their genetic variants. And as I said, initially, we could look at maybe 50,000 genetic variants. But now we can type at a higher resolution. So we can look at about 800,000 genetic variants in cases and controls. And then the statistics are relatively straightforward. It's a chi-square test to look, is a specific variant associated more with disease or health? And that was the basis of genome-wide association studies and still is. But what has changed is that with more and more of us organizing ourselves well, we've had bigger cohorts, which are better phenotyped clinically. Because of that, we can detect smaller changes. And also the technology of the platforms has improved. So where we had SNP typing, now we have whole genome sequencing, but the mechanism hasn't changed. So the really interesting thrombosis GWAS, the most recent two are one by the UK Biobank. And as a reminder, the UK Biobank had about 500,000 healthy volunteers there. And a part of these 500,000 would have gone on to develop a thrombosis. So in the UK Biobank, they tried to make a clinical risk score for the development of thrombosis. The biobank was also fully typed on an FV800K chip, but here we just look at the clinical risk to develop thrombosis, and they could make three categories: low, intermediate, high. And the difference between those categories was a fourfold. If you just look at the risk of developing thrombosis based on the risk factors as age, sex, BMI, previous cancer, smoking, and fracture risk. And these risk factors were picked because those were the most prominent in the analysis. If we do something similar for this, the variance, so out of the top GWAS variants of the association study. Out of the 102 top variants, we picked the 36 variants which were most indicative. And again, we made a stratification of low, intermediate, and high. And there you see the difference between the top and the bottom risk categories is a threefold. But the power is in combining it. So when you combine the clinical scoring, and why wouldn't you combine it, and the genetic scores, you can see that you can better stratify people who will develop VTE, and the difference between the highest and the lowest group is 7.5. So again, the combination of genetics and clinical data can help risk stratify, and I think that will clinically be very useful to further prevent thrombosis by picking those who would benefit from thromboprophylaxis. Genomicists like expanding. So after having done the UK Biobank GWAS, the Americans combined with the UK Biobank with their Million Veterans project. And by doing this combination, we suddenly, for the association study, had 26,000 cases and 624,000 controls. And the findings of these genome association studies were replicated in an astonishing 17,000 cases and 176,000 controls. Discovery and replication are standard in these studies. And the latest, the big paper in Nature Genetics, identified 33 loci, so 33 regions of the genome which are associated with thrombosis, of which 22 were new. Now, these loci each warrant further investigation, and that can take up to decades. However, where I find it clinically interesting is that you can do the same game. You can make another polygenic risk score. So this polygenic risk score is based on the Multi-Million Veterans Project and the UK Biobank. And let me just talk you through this, because people get scared, but actually it's not scary at all. So you see a Gaussian curve. You could have drawn any Gaussian curve for any trait, for height or possibly, but this Gaussian curve represents the polygenic risk score for VTE, to develop VTE. And as you can imagine, there will be extremes. Now, if you look at the highest group, the highest 5% of developing VTE based on the polygenic risk score, we can look in that section into more detail. So this highest group are those who either have a factor V Leiden variant, which we knew that that's something we accept, a prothrombin variant, and you can see the odds ratios in the bottom here. But also, what is quite interesting is if you make a combined stratification of the top scoring SNPs or variants, you get an odds ratio which is very comparable to those two. You can strengthen this risk score by making all kinds of combinations. So combinations of prothrombin variants with these SNPs, or the factor V Leiden with these SNPs. So why am I showing you this? I'm not showing you this because I think that's massive odds, but I'm just showing you that we can incorporate some of the state-of-the-art data from the GWAS studies to better predict risk. So that brings me to the current state of play in thrombosis and the genomics. So in red, you can see the kind of traditional genetic risk factors, which were all protein-based and coagulation cascade-based. But since then, I hope I've explained to you that we generated another 33 loci, which definitely need further study. Now, you can ask, where does this bring me clinically? And for that, I just want to take you back into one of the big genomic projects which have been led from the UK, and that was the rare disease pilot. And that was the start of the pilot for the 100,000 genomes. And in that pilot, over 13,000 participants partook from all over the world, and they all had whole genome sequencing. And the goal of this was to understand and find those rare disorders, but also to understand and learn how we would report variants in a clinical suitable way. and that arm was done by Thrombogenomics. So Thrombogenomics was again an international collaboration led very much by the UK, and people like Andrew Mumford, Professor Mumford, Professor Chowdhury, Dr. Gomez have been instrumental in this. So in Thrombogenomics, what was new is we tried to do a panel test versus testing a single gene for disease. And why would we do that? The benefit of doing a panel is that you can test multiple genes in one go rather than sequentially doing one gene. It becomes cheaper because if you have to do a gene every time, that's just quite time consuming, and it is more affordable. And through work done through that pilot, we now have NGS testing in clinical practice for hemostasis and thrombosis. The base of this Thrombogenomics platform, the capture, was that it would contain over 100 genes. It would contain all the protein coding regions, but also the beginning and the end bit of the genes, because those have regulatory elements, and all those areas which we thought would be implicated in disease. And this platform, it's kind of a virtual thing that changed over time. As I said, it's not only for clotting, so it has platelet genes, which are obviously involved in bleeding disorders, but it also has a list of prothrombotic genes. And this list is about 17. And these are the lists which were curated by also the I-State subcommittees as most important in clinical practice. So this is a beautiful tool from research to clinical practice, and we all have access to this panel. We validated the panel, so we looked at 2,396 patients with a bleeding or platelet disorder, and we looked at how the panel performed against their normal standard test. And if we focus on the thrombotic disorder, which was about 10% of this cohort, in about up to half, we were able to identify a variant. Some were pathogenic, so we know that they were causal. Some were likely pathogenic. They might be causal, but it needed more information. And there were also variants of unknown significance. But in a good half, we never found a variant. So this could be because it wasn't genetic or we weren't there yet. And I think that these panels should be incorporated in diagnostic pathways. And we do that on the other side, so on the bleeding and platelet disorders. And why do we do that? Because actually, it's a slightly simpler disorder. So some people bleed, some people have low platelet counts, and it can be underlying to a lot of different genetic variants. And you can see here that platelet disorders, sorry for the sidestep, is a multisystemic disorder. And we better know that certain platelet disorders, for instance, are result in renal problems or in cancers. And that's why we want to know what the underlying molecular basis is, because it impacts their treatment and management. So coming back to what do we now do with this R97 panel? So this is the NGS panel which is available. Now, the latest BSH guidelines written in 2022 do mention this panel, and they say, well, you can use it for thrombophilia with a likely monogenic cause. So it has to have clinical features. And as I spoke to you through the talk, you can see young age, spontaneous, very weak environmental risk factors. You could do this test, but it says very clearly, testing should only be used where it will impact clinical management. And that's the challenge if you follow the guideline. Because what do they say? What does the BSH guideline say on thrombophilia testing? Well, my summary is actually do not test. And I understand that with the clinical hat, but it's quite difficult with the scientific hat. But what they say is if you do a test, that should result in an effect for your patient. And just testing for the sake of it will not help anyone except for the scientific community. There are a few exemptions where they say asymptomatic first-degree relatives of probands with protein C, S, and antithrombin deficiency, where this might influence the management and life choices, can be tested. So, in an ideal world, we would have this beautiful loop where we do clinical data, genomic data, And if we know what the variant causes, by an MDT, we report back to the patient. And if we don't, we do genetic analysis. Unfortunately, in thrombosis research, we are not there yet. But I think we can be, and we should be, because where do I think that genomic fits into the picture? So I think it would be to discuss who to give thromboprophylaxis. As I showed you, we have much more evidence about risk by polygenic risk scores. We should think about which women in pregnancy and postnatal periods should have thromboprophylaxis. We have beautiful guidelines, but maybe we can expand on that. We can talk about tailored assessment of duration of treatment. How do we counsel young patients who've had an event? What is their likelihood of a re-event? And could that be based on a genetic cause? Should we talk about prevention in high-risk families? There's not much data yet, but we can gather that as a community. And what about additional phenotypes? I mean, why would platelet disorders be related to other phenotypes, but inherited thrombotic variants not? So, having said that, I just wanted to let you know that I hope I've convinced you about the need for further research, and we have ways of doing that. So the NIHR recruitment is open for young thrombosis. So if any of you have patients, and this is for scientific purposes, who are young, under the age of 30, with a family history of thrombosis, please do send them to me, because we can look in more detail. And what we do there is we look at all the clinical data, we do whole genome sequencing, RNA sequencing, proteomics, to align those things, because we believe that in a subset of the individuals where we can't find a monogenic cause with the standard panels, it was like 50% in the R97, it could be due to variations which cause changes in expression. So, coming to my end point, I think genomics research in thrombosis has recently resulted in useful things. The NGS panel, GWAS loci, which we can use for polygenic risk scores. There is a role for genomics in diagnostics of thrombosis. The R97 panel is accessible, and I think we should start adding some of these polygenic risk scores to those panels. And the recall study is open for recruitment. So do please send interesting cases to me, and I can talk to you about that. So without further ado, this work is obviously not just done by me, but a big panel. And I just specifically wanted to thank Keith Gomez for all the work he's done in thrombogenomics, Willem Ouwehand, who's been a lifelong mentor, but also new bloods such as Amar, who's in the left top corner, who's been really grateful in gathering these families with young thrombosis. Thank you very much.
Terrific, Suthesh, and you've managed to finish just about at the right time, which is brilliant. It's just starting, isn't it? The whole of thrombogenomics is opening up. Can we just be clear? You want patients, or you want their blood samples if they fit your criteria that you've talked about?
Yeah, so thank you, Beverley. So there are numerous NIHR sites, so contact me, because it might well be possible that there's a closer site than myself at the Royal London, and we can even organize home visits for those patients. So yeah, we can definitely talk about that. But
you've got funding, have you? This is a funded research project.
Yeah.
We're talking millions here, are we?
Yes, there is funding for patients to be even visited at home, to take their bloods, to get the genome sequencing done, proteomics done, RNA sequencing done. There's a whole team ready to go, and we've recruited our first patient. So funding, should it be a problem, I can sort all the logistical headaches with my team. But just if you have an interesting case, just drop me an email and I can guide you through that. And there is a lot of scope, Beverley, for research. So we gather some more data than we can actually do proper research on, as you can imagine. So if people are interested in doing research within the community, that's also absolutely fine. I'm not planning on sitting on the data myself. It's too much to handle. So anyone is welcome.
And is there a website we can look at?
Yes. So if you go to the NIHR recall study, but I can put that in my slides, which I will share with the people who were here.
Okay. Well, I have to wrap up now because we've got another session starting in 5 to 10 minutes. I want to say to the speakers, wow, thank you so much. I've learned such a lot from all three of you. And to tell everyone, if you came late or you had to leave early, this is going to be up on the Thrombosis UK website for you to see, perhaps for the first time, or just to revisit for the second time to catch up on all this amazing new information. So thank you all for fabulous talks, and let's hope we can do this again next year and have an update. Bye for now. Thank you.